Welcome to the deep dive. We're plunging into, well, one of the body's most critical yet maybe underappreciated systems today.
Absolutely. We're talking about internal waste management. It sounds mundane, but it's anything but. It's about survival, second by second.
Exactly. And the star player, the kidney, an organ of incredible efficiency, filtering and recovering with almost perfect precision.
It really is. Think about it. Every single thing the body does, metabolically speaking, creates byproducts. Some are harmless, like CO2 managed by the lungs, but others.
Others are seriously problematic if they build up. We're talking excess water, ions, and the big one, nitrogenous waste.
Get that balance wrong, and the whole system can go toxic very, very quickly.
So today, our conceptual deep dive, drawing straight from the NCERT material on excretory products and their elimination, is all about unpacking that genius.
We want to trace how animals, evolutionarily speaking, tackled this waste problem, especially nitrogen.
And culminate in the human kidney, how it manages that incredible feat of turning, 180 liters of fluid processed daily
A staggering 180 liters, yes.
into just about maybe 1.5 liters of actual urine.
Precisely. Nearly 99% recovery.
Yeah.
That's the core of its regulatory brilliance, and that's what we're aiming to understand. How does it do that?
Okay, let's start with the why. The waste itself. The NCERT is clear it's not just carbon dioxide and water.
No, you've got ions accumulating too. Things like sodium, potassium, chloride, phosphate, sulfate, either from metabolic reactions or just, taking in too much.
But the real challenge, physiologically, seems to be handling nitrogen.
That's the big one. It mainly comes from breaking down proteins and nucleic acids, catabolism.
And the NCERT points to 3 main forms this nitrogenous waste takes: ammonia, urea, and uric acid.
Correct. And the choice between these three isn't random. It's a fundamental evolutionary balancing act.
A trade-off.
A trade-off between how toxic the waste is and how much water it costs to get rid of it safely.
Okay, so let's take ammonia first. Sounds, well, potent.
Extremely potent, highly toxic, which means to eliminate it without poisoning yourself, you need a lot of water. Huge amounts for dilution.
So that strategy only really works if you're literally swimming in water.
Exactly. It's a strategy of ammonotelic organisms. The bony fishes, many aquatic amphibians, aquatic insects, they're surrounded by water.
And interestingly, the NCERT notes their kidneys aren't even the main players here.
Not for ammonia removal, no. Because ammonia is so soluble, it just diffuses out across their body surfaces, or for fish, across the gills, usually as ammonium ions and H4+.
So 0 water cost, essentially, because the environment provides an infinite buffer.
Right. But then life moves on to land.
And suddenly water is gold dust. You can't afford to lose vast amounts just diluting ammonia.
Precisely. So evolution needed a workaround, a less toxic form. Urea. This leads to ureotelism.
That's us, right? Mammals.
Us, yes. Also marine fishes, interestingly, and terrestrial amphibians. Urea is, get this, about 100,000 times less toxic than ammonia.
Wow. Okay, but there's no free lunch. What's the catch?
The catch is energy. The body produces ammonia first through metabolism. Then it has to invest energy primarily in the liver using the ornithine cycle to convert that toxic ammonia into the much safer urea.
Ah, so we spend energy in the liver to save precious water in the kidneys later on.
That's the core trade-off. Urea is soluble enough to travel in the blood safely and be filtered out by the kidneys, needing only a moderate amount of water.
And the NCERT mentions something clever some animals do with urea, retaining some of it.
Yes, a fantastic bit of biological tuning. Some animals, especially those in really dry places, deliberately keep some urea within the inner part of the kidney, the medulla.
Hold on, why keep waste? Seems counterintuitive.
To maintain osmolarity, basically to keep the inner kidney tissues really salty or concentrated. This high concentration gradient is absolutely vital for pulling water back out of the final collecting tubes later. It's a calculated move. Hold on to some waste to save even more water.
Okay, that's clever. And then the third strategy, the peak of water saving.
That would be uricotelism, the excretion of uric acid.
Who uses this?
Reptiles, birds, land snails, insects. Think creatures where water is extremely scarce or heavy.
And uric acid's advantage.
It's the least toxic of the three, and crucially, it's almost insoluble in water.
Meaning it doesn't need water for removal?
Practically none. It's eliminated as a sort of paste or even a dry pellet. Minimal water loss. Absolutely vital for birds in flight or desert dwellers.
So a clear progression. Ammonia needs loads of water, urea needs a moderate amount, uric acid needs almost none. driven by the environment.
Exactly, and that evolutionary path is reflected in the structures animals use. Before we get to our complex kidneys, the NCERT gives us a quick tour of simpler systems.
Right. Vertebrates have kidneys, but invertebrates use simpler, often tubulate forms, like protonephridia, flame cells.
Flame cells, yes. Great name, comes from the flickering cilia that drive fluid movement. You find these in platyhelminthes, flatworms like planaria, also rotifers, some annelids, and even the cephalochordate amphioxus.
And their main job, according to the NCERT,
Primarily osmoregulation, managing the internal balance of ions and fluid volume. Waste removal might happen too, but for many of these often aquatic organisms, keeping the fluid balance right is the #1 priority for these structures.
so fluid specialists first, then moving up a bit, we get nephridia.
Nephridia, yes.
Yeah.
More distinctly tubular structures. Think earthworms and other annelids. These are a bit more advanced.
How so?
They clearly handle both jobs well, removing nitrogenous wastes and maintaining that crucial fluid and ionic balance, a step towards a multitasking system.
Okay. And for insects, like the cockroach mentioned in the NCERT Malpighian tubules.
Malpighian tubules. Really neat design. They're these fine tubules branching off the digestive tract.
So they don't filter blood directly.
Not quite like our kidneys. They absorb solutes, water, and waste from the surrounding hemolymph and dump them into the gut. Then, the hindgut does the clever bit, reabsorbing water and essential ions, leaving the uric acid waste behind. Very efficient for small bodies conserving water.
And lastly, the NCERT mentions antennal glands, or green glands.
Yes, found in crustaceans like prawns. They perform the excretory function there. So you see this diversity, from simple flame cells to these tubules, all tackling the same core challenges. Get rid of toxins, keep the water balance right.
Right. Let's zoom in now on our own system, the human setup.
Okay, the main components. A pair of kidneys, naturally. Then the tubes carrying the urine away, the ureters, one from each kidney. These lead to the urinary bladder for storage.
And finally, the exit tube, the urethra.
Correct. Let's focus on the kidneys themselves first. Their location is quite specific.
Where exactly are they? Bean-shaped, reddish-brown.
Yep. Tucked away quite high up in the abdomen against the back wall, the dorsal inner wall. They're protected, sitting roughly between the level of the last thoracic vertebra, that's T12, and the third lumbar vertebra, L3.
They're not huge organs considering their workload. What are the NCERT dimensions?
About 10 to 12 centimeters long, 5 to 7 wide, and only 2 to 3 centimeters thick. And they weigh, on average, maybe 120 to 170 grams each. Quite compact.
Let's slice one open conceptually. What's inside? There's that notch.
The hilum, that's the indentation on the inner concave side. It's the gateway where the ureter exits and the renal artery, renal vein, and nerves go in and out.
And just inside the hilum.
You find this funnel-shaped space, the renal pelvis. It collects the urine before it heads down the ureter. And the pelvis has these little projections extending into it called calyces. Singular is calyx.
Looking at the kidney tissue itself, the NCERT describes distinct zones under the outer capsule.
Yes, there's a tough outer capsule providing protection. Then two main regions, the outer cortex, which looks sort of granular.
And the inner medulla, which looks more striped.
Exactly. The medulla isn't just one solid block, though. It's divided into these cone-shaped sections, the medullary pyramids. The pointy ends of these pyramids project into those calyces we mentioned.
And what connects these zones? I remember reading about columns of Bertini.
Right, the renal columns are columns of Bertini. These are basically extensions of the cortical tissue dipping down between the medullary pyramids. They provide structural support and pathways for blood vessels.
All this structure serves to house the real workhorses, the nephrons. Millions of them.
Nearly 1 million per kidney, according to the NCERT. These are the microscopic functional units, the engines of filtration and reabsorption.
Okay, break down the nephron for us. Two main parts.
Two main parts. First, the glomerulus. This is a dense little knot, a tuft of capillaries. Blood arrives via the afferent arteriole, a tiny branch off the renal artery.
And leaves.
Via the efferent arteriole, which is slightly narrower, that difference, helps build up pressure inside the glomerulus, which is key for filtration.
And this glomerulus isn't just floating there.
No, it's nestled inside a cup-like structure, the Bowman's capsule. It's double-walled. Together, the glomerulus plus the Bowman's capsule, that unit is called the Malpighian body, or renal corpuscle. That's filtration central.
Okay, so fluid gets filtered out of the glomerulus into the Bowman's capsule. Then where does it go?
It enters the second main part, the long, winding renal tubule.
Winding is right. What are the sections?
It starts immediately with a highly coiled section called the proximal convoluted tubule, or PCT.
Proximal meaning close to the start.
Exactly. Then the tubule straightens out and dips down into the medulla, forming a hairpin shape that's the loop of Henle, with a descending limb going down and an ascending limb coming back up.
Then it gets coiled again.
Yes, back up in the cortex, it forms another highly coiled region, the distal convoluted tubule, or DCT, distal meaning further away.
And where does the DCT lead?
The DCTs from many different nephrons all drain into a common, straight tube called the collecting duct.
So lots of nephrons share one collecting duct.
Correct. These collecting ducts run down through the medulla, passing through those medullary pyramids, and eventually open into the renal pelvis, delivering the final urine.
Now, the NCERT makes a point about different types of nephrons. It depends on that loop of Henle.
A very important distinction for function, yes. Most nephrons are cortical nephrons.
Meaning they stay mostly in the cortex.
Right. Their loop of Henle is short and only dips slightly, if at all, into the medulla. For these, that special blood vessel network, the vasa recta, is either absent or very reduced.
But then there are the others. Juxtamedullary nephrons, close to the medulla.
Exactly. These are the deep divers. They have very long loops of Henle that plunge deep into the salty medulla. These are crucial for concentrating urine.
And they do have that special blood vessel network?
Yes. Remember the efferent arteriole leaving the glomerulus. In these juxtamedullary nephrons, it forms the usual peritubular capillaries around the PCT and DCT, but it also gives rise to a thin, U-shaped vessel that runs right alongside the long loop of Henle. That is the vasa recta.
And that parallel U-shaped structure is key for the countercurrent system we'll get to later.
Absolutely essential. The vasa recta's structure and blood flow direction are critical for maintaining the concentration gradient in the medulla that the loop of Henle establishes.
Okay, anatomy covered. Let's get to the action. Urine formation. The NCERT breaks it into 3 main steps. Step 1, glomerular filtration, sometimes called ultrafiltration.
Right. This is the initial filtering step, and it's driven basically by blood pressure within those tiny glomerular capillaries.
The sheer volume here is astounding. What did the NCERT say?
Between 1,100 and 1,200 milliliters of blood flows through the kidneys every single minute. Think about that. It's about a fifth of all the blood your heart pumps out in that minute, constantly being presented for filtering.
And ultrafiltration implies it's a very fine filter. What makes up this filtration barrier?
It's a 3-layer structure, incredibly thin but effective. First, the wall of the capillary itself, the endothelium, it has small pores. Second, the inner lining of the Bowman's capsule, the epithelium. And third, sandwiched between them, is the basement membrane.
And those epithelial cells in Bowman's capsule are special, aren't they? Podocytes.
Podocytes? Yes. Foot cells. They have these intricate foot processes that wrap around the capillaries, leaving tiny gaps between them called filtration slits or slit pores.
So the fluid has to pass through the capillary pores, the basement membrane, and these filtration slits.
Correct. It's incredibly selective. Blood gets filtered so finely that almost everything in the plasma water, salts, glucose, amino acids, urea, passes through into the Bowman's capsule. The only things normally held back are the large plasma proteins and blood cells.
Which leads to this key measure, the glomerular filtration rate, the GFR.
Yes, the GFR. It's the amount of this filtrate formed per minute across both kidneys.
And the NCERT figure.
In a healthy person, it's about 125 milliliters per minute.
Okay, 125 millimeters. Now multiply that out.
And you get that astonishing figure. 180 liters of filtrate produced every single day.
180 liters. That's vastly more fluid than we even have in our bodies. This implies something crucial must happen next.
It implies that filtration cannot be the whole story. If we didn't reclaim almost all of that filtrate, we'd dehydrate and die incredibly quickly.
Which also means maintaining that GFR at the right level is critical. Too high, you lose too much. Too low, you don't filter waste effectively. How is it regulated?
The kidneys have a brilliant built-in regulator, the juxtaglomerular apparatus, or JGA.
Juxta meaning near the glomeruluses. Where is it?
It's a specialized little region formed where the distal convoluted tubule loops back and physically touches the afferent arteriole, supplying its own glomerulus.
Clever positioning.
Very. The cells in both the tubule wall, the macula densa, and the arteriole wall, the JG cells, are modified. They act as sensors. If the GFR drops, maybe due to low blood pressure, the JGA detects this.
And it responds.
The JT cells release an enzyme called renin. Renin kicks off a hormonal cascade that ultimately raises blood pressure and restores the GFR. It's a vital self-correction mechanism.
Okay, so filtration happens, but then comes step 2, reabsorption. This must be where the 99% recovery happens.
Exactly. This is the kidney's efficiency secret. Comparing the 180 liters filtered to the 1.5 liters or so of urine we actually produce tells you, yes, nearly 99% of the filtrate has to be reabsorbed back into the blood.
How does this happen? Who does the work?
The epithelial cells lining the renal tubule all along its length, from PCT to collecting duct, they reclaim substances the body needs.
Is it passive or active?
Both. Some things, like glucose and amino acids, and many ions like sodium, are actively transported back. This requires energy, ATP. Other things, like some nitrogenous wastes, and importantly water, especially early on, follow passively, often driven by osmotic gradients set up by the active transport of salts.
Okay, reclaim the good stuff. Then step 3, tubular secretion. This sounds like the opposite of reabsorption.
It is. Here, the tubular cells actively transport substances out of the blood from the peritubular capillaries into the filtrate flowing through the tubule.
Why do that? Why add more stuff to the fluid you're trying to get rid of?
It's about fine-tuning and getting rid of specific things the body really wants out, or things that weren't filtered effectively initially. Key examples are hydrogen ions for pH control, potassium ions, and ammonia.
So secretion is crucial for balancing ions and acid-base levels in the blood.
Absolutely vital. It's the final check, ensuring the blood chemistry stays within its very narrow, healthy range. Filtration, massive reabsorption, and selective secretion. Those are the 3 pillars of urine formation.
Right, let's follow that filtrate now, piece by piece, through the tubule to see where this reabsorption and secretion happens. First stop, after Bowman's capsule, the proximal convoluted tubule, PCT.
The PCT, this is the main reabsorption site. It's lined with simple cuboidal epithelial cells, but they have a special feature.
The brush border.
Exactly, a dense layer of microvilli on the surface facing the filtrate. This massively increases the surface area available for transport.
And what gets pulled back here?
A huge amount. The NCERT says nearly all the essential nutrients, glucose, amino acids, vitamins are recovered here, plus about 70 to 80% of the electrolytes, like sodium, potassium, chloride, and a similar percentage of the water.
So most of the bulk recovery happens right away in the PCT.
Yes, if the PCT wasn't working, the losses would be catastrophic. It also plays a big role, even this early, in pH balance.
Wow.
By actively secreting hydrogen ions and ammonia into the filtrate, and reabsorbing bicarbonate ions HCO3- back into the blood. It helps buffer the blood.
Okay, PTT handles the bulk, then the filtrate takes that plunge into the loop of Henle. This seems crucial for water balance.
Absolutely critical, especially for concentrating the urine. It works by creating and maintaining that high salt concentration, the high osmolarity, in the surrounding medullary tissue. The 2 limbs of the loop are key here, and they work differently.
The descending limb first, what's its property?
The descending limb is highly permeable to water, but almost impermeable to salts, electrolytes.
So as the filtrate travels down into the increasingly salty medulla,
Water is drawn out passively, by osmosis, from the tubule into the surrounding tissue. Salts stay in.
making the filtrate inside the tubule more and more concentrated.
Exactly. By the time it reaches the bottom of the hairpin turn, the filtrate is very concentrated, very hypertonic. Now it heads up the ascending limb.
And the properties flip.
Completely. The ascending limb is impermeable to water. Water cannot leave here, but it actively transports salts, mainly sodium chloride, NaCl, out of the filtrate and into the medullary tissue.
Actively pump salt out, but water can't follow.
Right, or sometimes passively in the thin segment. But the key is, salt leaves, water stays. So as the filtrate moves up the ascending limb, it becomes progressively more dilute again. And crucially, the salt pumped out here is what helps create that high concentration in the medulla in the first place.
A very clever system. Salt pumped out makes the medulla salty, which pulls water out of the descending limb. It feeds itself.
Precisely. It sets up the gradient. Now the diluted filtrate reaches the distal convoluted tubule, the DCT.
What happens here? More reabsorption.
Yes, but the NCERT calls it conditional reabsorption. This is where the fine-tuning happens, largely under hormonal control.
Conditional on what?
Conditional on the body's needs. For example, how much sodium and water are reabsorbed here depends on hormones like aldosterone and ADH, which we'll discuss. It's about adjusting the final balance.
And it also continues the pH regulation.
Yes, the DCT continues to reabsorb bicarbonate and selectively secrete hydrogen ions, potassium ions, and ammonia. It plays a key role in maintaining blood pH and that critical sodium-potassium balance.
Okay, almost there. The filtrate, now adjusted, moves into the collecting duct.
The collecting duct is the final checkpoint, especially for water. It passes down from the cortex, through the medulla where that salt gradient is strongest, towards the renal pelvis.
And its permeability can change.
Dramatically, yes, primarily under the influence of ADH. If the body needs to conserve water, ADH makes the collecting duct highly permeable to water. Large amounts of water then leave the duct, drawn out by that powerful medullary osmotic gradient, producing very concentrated urine.
And the NCERT mentions urea playing a role here too.
A small but significant one. The collecting duct allows a little bit of urea to pass out into the deep medullary interstitium. This urea actually contributes to maintaining that high osmolarity down there. It gets recycled partly through the loop.
So even urea contributes to water saving, and the collecting duct also does some final ion balancing.
Yes, it can secrete hydrogen and potassium ions as needed, further contributing to blood pH and ionic regulation.
This brings us to the overall mechanism that makes extreme concentration possible, the countercurrent mechanism.
This is the integrated function of the loop of Henle and the vasa recta working together. Remember, they run parallel, but the flow inside them is in opposite directions, countercurrent.
Filtrate goes down the descending limb, up the ascending limb. Blood goes down the descending vasa recta, up the ascending vasa recta.
Exactly. And this counterflow, combined with the specific permeabilities we discussed, is what establishes and maintains that incredible osmotic gradient in the medulla.
What are the numbers again for that gradient?
It increases steadily from about 300 millionths moles per liter on osmol, which is similar to normal blood plasma in the outer cortex, up to around 1,200 millionths ml in the very deepest part of the inner medulla.
A fourfold increase in concentration. And the NCERT says this gradient is mainly due to two substances.
Primarily sodium chloride, NaCl, and urea. NaCl is actively pumped out by the ascending limb, as we said. The vasa recta, acting as a countercurrent exchanger, prevents this salt from being washed away by the blood flow. It picks up salt going down and releases it going up.
And the urea recycling we just mentioned adds to that deep medullary concentration.
Yes, the combined effect of NaCl transport and urea trapping creates this powerful gradient.
And the payoff.
The payoff is that this gradient provides the strong osmotic pull needed to draw large amounts of water out of the collecting duct when ADH is present, allowing us to produce urine that can be up to 4 times more concentrated than our initial blood plasma. A phenomenal water conservation system.
So the kidney machinery is amazing, but it needs control signals. The NCERT highlights regulation by the hypothalamus, the JGA, and even the heart. Let's start with ADH.
Right, antidiuretic hormone, ADH, also called vasopressin. Its release is governed by osmoreceptors, mainly in the hypothalamus, that monitor blood concentration and volume.
So if you're dehydrated or your blood gets too salty...
And these osmoreceptors detect the change and signal the hypothalamus, which then tells the posterior pituitary gland, neural hypophysis, to release ADH into the bloodstream.
And ADH's target in the kidney.
It primarily acts on the later parts of the tubule, the DCT, and especially the collecting duct. It makes their walls much more permeable to water.
So more water gets reabsorbed back into the body from the filtrate?
Exactly. Less water is lost in the urine. It prevents diuresis, excessive urination, hence the name antidiuretic hormone. It helps you conserve water when you need it.
The NCERT also mentions another effect of ADH, vasoconstriction.
Yes. At higher concentrations, ADH also causes blood vessels to constrict. This increases blood pressure, which can indirectly help maintain the GFR if blood volume is low.
Okay, that's the main water balance hormone.
Right.
What about regulating blood pressure and GFR more directly, especially if they drop significantly? That JGA system again.
Yes, the renin-angiotensin mechanism, sometimes called the renin-angiotensin-aldosterone system, or RAAS. This is a more complex, powerful response.
Trigger by
A fall in glomerular blood flow, glomerular blood pressure, or GFR itself, detected by those JG cells in the juxtaglomerular apparatus.
And they release renin. What does renin do?
Renin isn't a hormone itself, it's an enzyme. It circulates in the blood and acts on a plasma protein produced by the liver called angiotensin. Renin converts it into angiotensin I.
Is angiotensin I the active player?
Not yet. Angiotensin theta is then converted into angiotensin II, mainly by an enzyme called ACE, angiotensin converting enzyme, found largely in the lungs. Angiotensin II is the really potent one.
What does angiotensin II do?
2 main things, both aimed at raising blood pressure and GFR. First, it's a very powerful vasoconstrictor. It tightens blood vessels throughout the body, directly increasing blood pressure.
Okay, direct pressure increase. And the second thing?
It stimulates the adrenal cortex, the outer part of the adrenal gland sitting on top of the kidney, to release another hormone, aldosterone.
Ah, so renin leads to angiotensin II, which leads to aldosterone. What's aldosterone's job?
Aldosterone acts specifically on the distal parts of the nephron tubule, DCT and collecting duct. It promotes the reabsorption of sodium ions, Na+, from the filtrate back into the blood.
And where sodium goes?
Water tends to follow, by osmosis. So aldosterone causes sodium retention and consequently water retention. This increases blood volume, which further helps raise blood pressure and restore GFR.
So RAAS is a multi-pronged system. Vasoconstriction plus salt and water retention. Very effective at raising low blood pressure.
Extremely effective. But you need a counterbalance, right? You don't want blood pressure to overshoot.
The NCERT mentions a check mechanism from the heart.
Yes, atrial natriuretic factor, or ANF. When the atria, the upper chambers of the heart, are stretched by increased blood flow or pressure.
Meaning blood pressure is getting too high.
Exactly. The atrial walls release ANF, and ANF does pretty much the opposite of the RAAS.
How so?
It causes vasodilation, widening of blood vessels, which lowers blood pressure. It also inhibits renin release and aldosterone secretion, promoting sodium and water loss. So ANF acts as a natural brake on the renin-angiotensin system, keeping blood pressure in check.
A beautiful system of checks and balances. Okay, the urine is formed, concentrated, balanced. Now, how do we get rid of it? Micturition.
Micturition, yes, urination. The urine flows down the ureters and is stored temporarily in the urinary bladder.
Which can stretch quite a bit.
It can. As it fills with urine, the bladder wall stretches. This stretch activates receptors in the wall.
Stretch receptors send a signal.
They send nerve signals up to the central nervous system, the CNS. The CNS processes this and, when appropriate, sends motor signals back down.
What do the motor signals do?
They cause two things to happen. The smooth muscle in the bladder wall contracts, squeezing the bladder, and the urethral sphincter, the muscle controlling the exit from the bladder, relaxes.
Contraction plus relaxation equals...
Release of urine. That coordinated action is the micturition reflex, though we do learn voluntary control over the external sphincter.
And typical volumes, the NCERT gives figures?
Yeah, an adult human usually excretes about 1 to 1.5 liters of urine per day. It's typically light yellow, slightly acidic around pH 6.0, and the main nitrogenous waste is urea, about 25 to 30 grams excreted daily on average.
And analyzing this urine is medically important.
Hugely important. It's a window into metabolic health. The NCERT gives examples. Finding glucose in the urine, glycosuria, or ketone bodies, ketonuria, are strong indicators of conditions like diabetes mellitus, where blood sugar regulation is impaired.
Well, the kidneys are the stars of nitrogenous waste removal and fluid balance. The NCERT rightly points out other organs play supporting excretory roles.
Absolutely. The lungs, for instance. We often think of them just for breathing,
Yeah.
but they're massive excretory organs for carbon dioxide.
How much CO2?
Around 200 milliliters per minute. That's a huge amount of metabolic waste gas. Plus, they eliminate significant amounts of water vapor with every breath.
Then there's the liver. We mentioned it makes urea, but it eliminates other things too.
Yes, the liver acts like a processing plant. It secretes bile, which contains substances it wants to get rid of breakdown products of red blood cells like bilirubin and biliverdin, which give bile and feces their color, excess cholesterol, degraded steroid hormones, certain vitamins, and many drugs.
And these bile components leave how?
They enter the digestive tract with the bile and are eliminated from the body along with digestive wastes in the feces.
Okay, lungs, liver. What about the skin?
The skin contributes in two ways. Firstly, through sweat glands. Sweat is mainly water and salt, sodium chloride for cooling, but it does contain small amounts of urea and lactic acid, so it's a minor route for nitrogenous waste.
And secondly?
Through sebaceous glands. These produce sebum, the oily substance that protects skin and hair. Sebum also eliminates certain substances like sterols, hydrocarbons, and waxes.
And the NCERT even mentions saliva.
Yes, a very minor point, but small amounts of nitrogenous waste can apparently be eliminated via saliva too. So excretion is a team effort, though kidneys handle the critical water-salt-nitrogen balance.
Now, what happens when this critical kidney function fails? Uremia.
Uremia is the condition where urea accumulates to toxic levels in the blood because the kidneys aren't filtering it out effectively. It's a sign of serious kidney malfunction and can lead to kidney failure if not treated.
And the main treatment mentioned is hemodialysis, the artificial kidney.
Yes. Hemodialysis is a lifesaver for people with kidney failure. It essentially does the kidney's filtering job externally.
How does it work physically?
Blood is drawn from an artery, usually in the arm, and heparin, an anticoagulant, is added to prevent clotting. This blood is then pumped through a special filter, the dialyzer.
What's special about the dialyzer?
Inside, the blood flows through thin fibers or channels made of a semi-permeable membrane, like cellophane. Think of it like thousands of tiny filtration tubes. These tubes are bathed in a special fluid called dialyzing fluid or dialysate.
And the composition of this fluid is key.
Absolutely critical. The dialysate has the same concentration of normal plasma components, electrolytes, glucose, etc., as healthy blood. But crucially, it contains no urea or other waste products.
So the waste moves out by diffusion.
Exactly. Because of the concentration gradient, urea, excess potassium, excess phosphate, and other wastes diffuse out of the blood across the membrane and into the dialysate, which is constantly being refreshed. Excess fluid can also be pulled off.
And the cleansed blood
Is then returned to the patient's body through a vein after adding an anti-heparin agent to restore normal clotting. It's a temporary fix, often needed several times a week.
But for permanent failure, the NCERT points to transplantation.
Kidney transplantation is considered the ultimate treatment for end-stage renal failure. It involves surgically placing a healthy kidney from a donor into the patient's body.
Donor matching is important.
Very. Preferably a close relative to minimize the chances of the recipient's immune system rejecting the new kidney. Immunosuppressant drugs are also needed long-term.
Okay, and just quickly, 2 other common kidney problems mentioned: renal calculi.
Renal calculi kidney stones. These are hard, insoluble masses formed from crystallized salts, often oxalates or calcium phosphate, within the kidney or urinary tract. Can be extremely painful.
And glomerulonephritis.
That's inflammation of the glomeruli, those filtration units. If the glomeruli get inflamed and damaged, filtration is impaired, proteins in blood cells might leak into the urine, and kidney function declines. Hashtag outro. You know, looking back across everything we've discussed from the NCERT, it's not just the scale of filtration or the complexity of the tubules that's impressive. It's the sheer elegance of the control systems.
The feedback loops. JGA, ADH, ANF.
Exactly. The body is constantly self-monitoring, adjusting pressures, permeabilities, ion transport, all to maintain that incredibly stable internal environment, that homeostasis. The kidney isn't just a filter, it's a master regulator.
It really is remarkable taking that initial massive 180-liter filtrate and sculpting it through precise reabsorption and secretion into just that final, carefully balanced 1.5 liters of waste. It feels almost like biological alchemy.
It's an incredible feat of engineering, honed by millions of years of evolution. And maybe, as a final thought for you listening, consider that evolutionary pressure again. Why did life on land invest so much energy in converting a relatively simple ammonia into more complex urea or uric acid?
The water cost.
The water cost. It shows how biological adaptation, as laid out in the NCERT, will often prioritize survival, in this case, water conservation, even if it means using a less direct or more energetically expensive biochemical pathway. A fascinating compromise, essential for colonizing the land.
A profound point to ponder. Well, that's all we have time for on this deep dive.
Thank you for joining with us today. See you next time.